the drug discovery, the drug discovery process is such challenging process. So it's like a movie, when you think of a movie, if the villains are strong, the hero needs to be super strong, right? And throughout his life, he has done a lot of research exercise to make him a robust person. That's where we stand in front of diseases. We, as a scientist, as a contributor to the field, we need to stand with resilience so that we can tackle the problem, right? Think about the pandemic, how we tackled as a human mankind. So you have to be very resilient, passionate, and very hardworking if you want to be in this area. Okay. So medicines are boon to mankind. It goes without saying, होइन. So like यो big miracle जति पनि भएको छ आजको वर्ल्डमा, स्पेसली हेल्थ केयरमा. So मेडिसिनको एकदम इम्पोर्टेन्ट रोल छ, होइन. So it's a boon. And there are a lot of medicines that are still required. So we need people like you in the future to come up and be a part of the journey. Okay. Okay, let's do this. What do you think is the hardest part of the medicine? Was all of the above? Have you ever thought this? So when you think of a funnel, right? So I always think this drug discovery and development process is a funnel where on the top you put a lot of things, which is high throughput screening compound. If you want to go to a virtual library, there are billions of compounds. At the end of the day, and you are going to make one pill. So what happens when the compound is coming down from the top of the funnel is an important question, right? So what are the choices made in the process? Why millions of compounds fail and only one go in reach? They are always made on a rational decision, and each rational decision is guided by deep scientific innovation, right? So किन हुन्छ त यो भन्ने कुरा जान्नु एकदम इम्पोर्टेन्ट छ. Okay, so you fuel with a billion things and you just get one, the best, that is treating a disease. So who are the villains now? In the movie Drug Discovery, who are the villains? This is a global health. And do you know how many diseases exist in the world? I was trying to research this and try to come with the data. There is no number actually. So disease exceeds millions. So there are a million of things that can bother you guys, that can bother humanity, that can bother health sector. So all of these are villains. And to combat these villains, we need to come with a pill, with something that you treat it, right? So there is a humongous amount of burden in humanity to combat with the challenges that are associated with disease. If we think with that lens, right? Millions of people die due to diseases. Isn't that shocking? And I'm very happy to see that you guys are in such a wonderful field where you can contribute to the human mankind towards developing therapeutic agents for different diseases, right? So I hope you'll join this venture, this nice scientific journey of combating diseases. So before I talk about disease, I want to praise two things to everyone. First is just don't develop a lifestyle where you don't want to take a pill. So first philosophy you need to bring. Even I am a student of medicine, medicinal chemistry, pharmaceutical sciences, pharmacology. I just don't want people to come and eat the pill. I want them to be healthy, right? So it's a backup option. So the first thing you need to maintain is a healthy lifestyle. To undergraduates who is doing bachelor's, I don't think I need to explain what healthy lifestyle is. That goes without saying, right? Prophylaxis and vaccination. The second is even you are before you are ill, there are ways your body has known, there are ways the world has learned that there can be future attack on you, right? Tuberculosis can be one, polio can be other, and there are a lot of prophylaxis and vaccines that are already developed, and you are already administered too, I'm sure. So that should be our second options. So treatment and disease management is our third options, right? So before thinking about treatment, just think about preventing from the disease itself. And the main strategy are healthy lifestyle and willingness to undergo prophylaxis, vaccination, right? Okay, now we are here on the main topic, I guess. So drug discovery is a scientific process. There are going to be good cops, the bad cops, and the ugly cops. So It's not an easy journey, otherwise everyone would make a pill, right? It's a very hard journey. It needs a combination of brain and input from all the talent minds in the world. It needs a deeper understanding of everything, because at the end of the day the pill should be able to resolve the disease, and that's not easy, right? So, as Nature Review on Drug Discovery in 2008 correctly pointed it out, like it took 12 to 15 years. So that might be decreased now with the advent of AI and machine learning. There can be a lot of things we can take their help. So the time is likely to come down in the near future. However, still 10 years is a very long time, right? There is a disease and you can only find the treatment for that disease after 10 years. It's a very long time, right? So it's $1 billion investment, right? So a lot of attrition. As I said, a company starts with a billion of compound. If the company gets lucky, there is one pill that comes into market, but the attrition is more than 70%, which means they get zero pill when they fuel with the funnel. So they are technically wasting billions of dollars. And the sooner the fail happen, it's good, because when it goes to clinical trial, the phases are going to be super duper expensive. So at that point, early failure is better than late failure, right? So there's a chat. Let's see. Okay, so target discovery, identification, and validation is the first step, right? So in what happened in target identification? So that's the crucial step in identifying drugs, right? So every disease has its own pathogenesis. So a disease is a process where the body's mechanism has changed or altered. So there is a target that is overexpressed, that is underexpressed. It's beyond normal. Now we need to be able to harness that target. So finding a target is the first challenge, right? So there are different ways to do that. So for example, you can take a molecular approach or systematic approach, where you can take the patient samples, send it for next generation sequencing, and try to see what are the genes that are overexpressed, and then try to identify what are the proteins that are encoded by those genes, or try to find the protein that are overexpressed, right? There are ways we can use proteomics and genomics to find a target. So once you find a target for a particular diseases, the second important thing is to ask, is that target druggable? Which means if a target is given, can the drug goes and bind to the target, and that will kill the disease? So again, a concept of genomics and proteomics are used. The most common that is trusted well in the industry is knockout strategy, right? So for example, think this. So if a person has a cancer, it is highly likely that the person has overexpressed a cancer gene. That cancer gene is responsible for a protein that would eventually lead to cancer. Now, if we can genetically knock out that gene, so that protein, so protein is encoded by DNA, right? That's gene, right? So if you can knock out that, you can knock that protein out. So what does that do is, if you knock out a protein, so in those genetic knockout models, they shouldn't have cancer. That's how they are validated by genetic methods. There are also molecular methods. So you induce cancer, and then you knock out that gene, and you say, okay, the cancer is gone or cancer come up. So there are ways to validate, but validation is very important, because nobody wants to spend a lot of money on a target if the target is not valid. So it's establishing a robust mechanism to establish, if we can target X, the disease will be cured, is so important. Okay, so this go in iteration. So first thing in modern drug discovery is identification of target and validation of the target. Okay, so there was a question sent to me that says how to design drugs for a specific disease. I think it's a point here to answer this. So for a specific disease, you want to design a drug, you want to identify a target. So for that, you need to understand the pathogenesis of a disease. So for example, if you're talking about virus, what is a factor in the virus that can be selectively killed without killing the human cells? If you are talking about some signaling issues in our body, then we need to figure it out. It's what is the mechanism that is taking part. So it's a unique question, and the answer would be depending on what type of problem you are trying to solve. At the end of the day, you have to target a gene, a protein, or something, right, at the receptor level, ion channels. So there are a lot of targets that are established. Once you find that target for your disease, you have to do the things that I'm going to explain in the further slide. But identification of target is very important, and each target is unique to a disease. So the most important input to design a treatment for a specific disease is actually to find the target. Now, after this, different drugs work on the same target. So if you are finding an inhibitor, that will work on the same target. There could be multiple scaffold that can be acting, but identifying the specific target for the specific disease is very important, right? So once you identify target, you need to establish a quantitative way to assess these values. I'm pretty sure as a student of pharmaceutical sciences, you might have word like IC50 values, right? Ka, Ki, Kd, right? You have frequently heard these terms. Yeah, these are the terms that are used to quantitatively express the binding, the killing of the cells. So depending on what terms you are using, they mean different thing. At the end of the day, you have biochemical assay, which talks about isolated protein, and your compound of interest is going to be. Hi, is that
I would like to request Sanjay Adhikari to please mute his mic.
Oh, okay, sorry. I was thinking there was a person. No worries. So in the biochemical assay, an isolated protein is going to be targeted by the drug of interest. So in this case, we call chemical entities, compounds, molecule, because they are not drugged, right? So we measure the affinity of binding, like how powerful those bindings are. It can be also done as a phenotypic way. For example, you culture a cell compound and you see how many cells died, and then you can plot that against the concentration. So that there are many quantitative ways, but to establish that your compound is better for the target, there should be always a assay that is robust, that is reproducible, and this assay can be at a biochemical level, at cellular level, or even at organoid level, but there should be established quantitative metrics. And people spend a lot of time trying to identify an assay for a target, because now you go to a high throughput screening setting, right? You like most of the pharmaceutical company has compounds in their library. So the company I'm currently working with, AbbVie, has 1.5 million compounds in their library. So they will use their library to identify the first compound that is going to go and bind. And then once the hit is identified, the project begins. Okay. So heat identification and heat to lead optimization. Now this leads to me about a new concept, right? Is where most of the medicinal chemists like us are involved. So as a medicinal chemist, we need to understand the data provided to us by the molecular biologists that talks about target identification and target validation. Looking at those data, we should be able to interpret, are we going to pursue a drug development program in targeting this target for the disease. After we are convinced the data, the next thing we have is to do heat identification. Now, it's very difficult to find the heat because anything can go and bind in that target, right? So there are two ways people take. So there was a question too that was saying, how about computational and AI, right? So yes, with computational, there are billion of compound. If you want to list, there are two libraries that I pretty much go to. They are called enamine library and Zinc database. So they have 6 to 7 billion compounds, and they can be docked in a platform. There are free platforms too. I use Schrodinger, but there are many others that can be used. And when you dock those compounds, the crystal structure is freely available in PDB. You pull the data, you dock the compound, you find the best hit. That's how computational approach do, right? And how is AI helping? Well, if you have to do 7 billion, it used to take 6 months of computational power. But now what AI does is that trains the data set. For example, first 1 billion in cluster so that the work can be done in a couple of days. And now it can build more on the scaffold. So AI is really taking over this computational lead driven tool. And there was a question on that too. However, there is another way too. So the other way is high throughput. So the library that was developed can be used to actually bind. Okay. So one thing with computational work, there is always a doubt that that should be backed up by experimental claims. So if a computational tool gives you a value, you have to do that in lab to test if that data is right. Okay. So there is always a caveat to that. However, if you do experiments, your confidence level is higher. So this is atorvastatin. So you have to know that every molecule you eat is a chemical substances, and as a medicinal chemist, you have to think about what each functional group. So this benzene ring, this amide, this benzene, this fluorobenzene, this 2-hydroxyl group, carboxylic acid, what that is helping, what is that doing in the enzyme pocket. It's very important to rationalize that. Okay, what type of interactions are important? What's the size? What's polar surface area? What is TPSA? What is the log P of that proper compound? Is it bioavailable? How can you predict? So as a medicinal chemist, the structure says a lot of things. Okay, what are the type of interaction in the pocket? What amino acids there could be? So and as a person in the pharmaceutical field, I think it's a good idea to have to look at structure and try to understand its 3D, try to understand in its in pocket, right? So imagine if you could build a drug like a superhero. What power would it need to fight the disease? Right? This is where medicinal chemists work a lot to try to find the best lead compound. And this cycle is very iterative. Okay. So for example, you screen a million of compound, and for example, you get 10. So now with 10 compound, you make a small library, and then you make 10 into 5, like 50 compounds each on the library, right? And then you keep on optimizing. Again, so there are 3D QSAR model, which means it can be done computationally too. Focused library design, structure guided optimization, and this mapping where you contour plots and try to understand the trends, right? So the most important thing for a lead candidate is they should have excellent ADMET profile, which means, right, ADME. So genotox assessment, so it should be safe. So safety and efficacy should be there, but also good pharmacokinetic profile of the compound is equally important. So we, as a medicinal chemist, or as a person who are involved in the drug discovery and development, should constantly think and ask this question. Okay, is my compound binding properly to the target? Because that's the first question we need to address. The second question we need to address is does it have good ADMET profile? So before we go to in vivo studies, ADMET profile tell a lot of things, right? How is the metabolism? If you incubate your compound in the human liver microsome, how much time it lasts tells you a lot about the metabolism profile, right? It tells a lot about Cmax, Tmax, a lot of pharmacokinetic profile, right? That you might have learned in pharmacology. So toxicity assessment is another important criteria. A compound, whatever powerful, whatever it could do, should be always safe to use. So that's rule number one in drug design is you can't a compound that is either genotoxic, that is mutagen, or that has serious side effects. Okay. So that's where we need to balance. Now, as a chemist, it's always a question about what structure is highly electrophilic or highly nucleophilic that the compound in the body is attacking and causing detrimental effects. How can you twin that out? Do you pull the electron density out? Do you supply electron density out? So in a structure, there is a lot of things that we need to understand this data. Something is toxic, we need to understand how, we need to tune it down. If the toxicity is so high, we should kill the program and move on to another scaffold, okay? So at the end of the day, we need a compound ready to go for the in vivo studies. After we assess that, we want to test this in real animals and animal models, okay? Okay, so there are a couple of questions here. What are key challenges in modern drug design? And these questions were asked by you guys, okay? Thank you for bringing this up. It provided me an opportunity to answer your question. My main intention भनेको तिमीहरूलाई help गर्ने हो हैन? So these questions really guided me to design what should I say, okay? Thank you for that. So the question was, what are the key challenges in modern drug design and how are computational methods helping to overcome them? I think I answered this. So the major challenges in modern drug design is absolutely time. So that is very big, and computational tool will, I think, ultimately help us reduce that time. The second is there is a very high rate of attrition, and the data, so the rational data for what would work and what would not work is very less. So I think with AI, if we keep on training, there should be a clear trend that we can understand these are very bad things, these would not work. So I think with computational, of course, the things are getting faster, but the field hasn't really absorbed it and got a real benefit till now. Hopefully, in a very near future, we can start seeing that, at least in the computational side, like docking, MMGBSA calculation, protein binding, the effect has been seen, and I think it would be good there too. Okay. The next question was, what are some key challenges in translating a drug from the research brains to clinical application, and how can medicinal chemistry help overcome them? Yeah. So there are challenges in drug design is everywhere. It could be target identification, it could be target validation, it could be lack of identification of a hit compound, it could be a lot of problems associated with the lead compound, it could be metabolic instability. So there are many challenges. Also, the challenge can come when it hits phase 2 of clinical trials. So the patient are there, but because human profiles and the experimental animals profile are very different, the efficacy in the human is so low. So now, as a medicinal chemist, you should be able to see these data. Medicinal chemists mainly work on the drug discovery and design part, but they should be able to see that data and say, it's toxic. So the toxicity is due to this. It is less efficacious because the bioavailability was poor. The log P profile of the compound was not good. So you go back to your structure and ask that question of why it failed, right? And based on that, they try to answer. I hope my answer was very vague, because the problem, if the problem is specific, medchem strategy could be applied. But if the problem is general, we should be ready to tackle in what problem we are thrown out. Okay, so that's a very routine problem in our group discussion session. Every week we discuss with a problem of attrition in drug discovery and development, and what would you do as a med chemist to overcome that. But great question, I appreciate it. Okay, so I told admit are important, but in vivo PK/PD profile, which means pharmacokinetic pharmacodynamic profile in the experimental animal, is equally important, right? So a drug, when you take it by any mode of administration, that's fine. It should go bind to the target, stay there for sufficient time, exerts its therapeutic effect, come out of the target, get eliminated from the body, right? Hit Cmax, in Tmax, come out of the body and get thrown out. Now, after 24 hours, there should be literally no drug in your body. So that's an ideal drug. Now, even though in the in vivo models, or even though in the, there are questions, let me see. So there are questions I'll address at the end, but that's, thank you for the questions. Okay, I like those. So in vivo PK/PD is very important criteria to assess during the drug development. So even you have great data assessing that the compound is bind super well, the compound is non-toxic when tested across many panel of cell lines, go and bind to your target. It has to be stable in your gut, it has to be stable in your stomach, it has to be stable in your intestine, it has to be stable in the blood, it has to go bind the target, it has to have sufficient residence time in the target, it has to change the signaling pattern, exert its effect, and get metabolized under phase 1 and phase 2 condition, right? A compound should do everything besides efficacy and safety. So we need to establish that the compound has all these properties. Now, the good thing is a lot of things are known about this, right? So we need to know, okay, what is the way we bind the compound, and then we do a lot of these experiments and try to assess the limitation and try to fix bit by bit. Okay, however, the main challenge is this here. If you want to solve a metabolic stability issue, right? The established issue is, so you want to identify what those metabolites are. You want to identify what cytochrome P450 isoforms were involved in the metabolism of that drug. Once you identify that, they are called metabolic soft spots, right? So you go and put a electron withdrawing group, for example, trifluoromethyl. And now you hope that the compound become metabolically resistant. However, the compound can also change its properties now, can change its efficacy, can change bioavailability. So changing one parameter can change other parameters too. So the same test has to be repeated once you decide to make that decision. Okay, so your activity shouldn't be compromised at cost of one. So it's all about a very good balance of a lot of things. Hence, drug design is difficult. Okay, so lastly, once you establish that the compound follows a good pharmacokinetic and pharmacodynamic rhythm, you go for assessing in vivo efficacy and in vivo safety, right? Though there seems to be last, but they are the most important thing a compound should have. It should go and exert its therapeutic effect. For example, an anti-cancer drug, to find if it is working, you need to induce cancer in mouse model. You need to feed that mouse for 14 days, dose that mouse with a compound, and see the effect over time that the tumor size is reduced or not, or stronger or not. And the same goes with different disease model, right? So there are disease model you need to create, you need to provide a therapeutic intervention, and you need to treat it. Okay, so that's how it has to be efficacious. And now in terms of safety, there should be no serious issues. So is it tumorigenicity, genetic stability, distribution, toxicology, a lot of side effects, adverse effects, drug interactions, you name it, you know a lot, right? So my goal today was not to go to any topic very deep, but rather stay on the theme of drug discovery and development and walk you through. Okay, so and if you want to, every topic is very deep in its own kind, and if you have research spark, kindly go and read. So there was a question here. How would you design a selective inhibitor for a specific isoform of kinase? Okay, so he might be talking about, so the kinase has different isoforms. Kinase is there are more than 100 kinases in our body, right? And they put phosphatase onto the structure. So OS reacts with phosphatase, and these reactions are done by kinases, ensuring minimal off-target effects. So because there are a lot of kinases in the body, so when you are trying to design a compound that target kinase, that can go and target other kinase. So there is always a risk, and even we talk this topic very frequently, that once you want to target something like this, which have more than 100 isoforms, targeting can be challenging. So explain the key strategy you would use, including computational and experimental approaches. Okay, it looks like a very exciting and interesting question. Whoever has asked this, yeah, it's a very great question, and it's a pretty common problem in drug discovery and development when you have to deal with any target that is multiple isoform. The main reason is the gene, the genetic makeup, and the protein makeup so a protein makeup is the amino acid sequence are very closely aligned. Therefore, they behave very similarly. However, how would we address this? Now, to address this, there are some questions here. Okay, we'll talk about this later. Okay. So so when the alignment is matching, still there are two different isoforms, right? So there might be something in the protein that is different. So if even it matches 95%, there might be 5% difference in the genetic makeup, or if you are thinking about a protein, the sequence of the amino acid. Now, that's how scientists brilliantly use that tactics to find the different active site or allosteric site on the protein to account for those differences. Now, sometimes you can target same thing, but the compound goes and bind to one isoform, but not the other. The reason is the pocket. So because they don't align, so the path to the compound, which is called pocket, is different in different proteins. So it's tricky. It's always a challenge, and sometimes you can't make a one isoform that is 1000x selective over other. You have to live with the reality that 10x is good enough. There will be some side effects, but we have to deal with it. So people also move with that part when you can't reduce side effect by more than 10x. Okay, so a great question. So the key strategy we are using, as I said, is try to bias why those isoforms are different and try to understand that from different perspective in terms of protein alignment, in terms of gene alignment, in terms of topography, in terms of the space it occupied. So there are a lot of things that differ when isoforms are differ, and then you use that, we try to rationalize that and try to design compounds. I hope I did okay job trying to answer this. Okay, so there are two type of people that come that who play a huge role in drug discovery and development. They are process chemists. So think of this, okay? A medicinal chemist in drug design and development make 5 milligram of compound, and that can test everything they want, right? Now, when you think of clinical trials, think of the number, thousands of people, right? When thousands of people are involved in a trial and all of them have to be given compound, it has to be produced in kilograms. And when it goes to real market, it has to be produced in tons, right? So to produce tons of compound, you need a team called process chemists. So their main goal is to apply the principles of fundamental synthetic chemistry principle and apply to making tons and tons of compound. It's a very challenging job. And they are very multi-step synthesis. There is a lot that I want to address. And this is also a job opportunities for you guys in the future. Process chemists think synthetic chemistry problem very deeply. They mainly work hard to meet global requirement of drugs, right? So the early things that I told were about testing, trying to identify drugs. The medicinal chemist like us can make those small amount of compound. But when it comes to make a global supply, we can't. We fail in our effort because there are different techniques and the expertise that they have had, process chemists, and I want to value their effort in the aspects of drug discovery and development. The other valuable scientist that drug discovery and development should absolutely pivot and respect to are formulation scientists. So formulation scientists deliver the most convenient dosage form, right? So what medicinal chemists do is try to find the active pharmaceutical ingredient that is administered to do a certain therapeutic effect. तर के गर्नु त्यो कसरी जहाँ पुग्नु पर्ने हो त्यहाँ पुगाउन सकिन भने that doesn't have any value, right? So त्यसलाई गर्न, okay, I have a question for you guys. Which is the most convenient form of dosage form? सबसे सजिलो के हुन्छ? सुई हान्न हुन्छ कि सोल्युसन खान हुन्छ कि के हुन्छ? सबसे नाकबाट हाल्न हुन्छ कि ओरल ट्याब्लेट खान हुन्छ? What's the easiest form you guys think?
Oral tablet.
Yeah, very good, right? So every company wants to make a pill that is oral tablet. Also, अचलको रिक्वायरमेन्ट तिमीले थाहा छ? Once a day, मान्छेले दिउँसो बिर्सिन सक्छ. So once a day, टक्क खाना खानुभन्दा अगाडि या खाना खाएर टक्क खायो, भोलिपल्ट फेरि अर्को खानु पर्यो. So that's the ideal goal that in pharma we want to address. So त्यसको लागि चाहिँ हामीलाई टी-हाफ लगभग 13-14 घण्टा चाहियो हैन? 10 घण्टादेखि 14 घण्टा भयो भने once a day would be fine. Okay, that's just a question there. So molecular, so there are a lot of formulation. मैले एकदम summarize गर्न खोजेको मात्रै हैन, and I have 1 hour and it's a lot to talk, so मैले summarize गर्न खोजेको. So there are different aspects this formulation chemist think हैन कहिलेकाहीँ. So we need to talk to formulation chemist. So when you have a pill, we have no clue how we are going to administer. अब यो stable हुन्छ हुन्न, tablet बनाउने, capsule बनाउने, solution बनाउने, we have no clue because we just think about API, GABA बनाएर चाहिँ माउसलाई दियो हैन. But this is a big problem, right? When a human take it, there are a lot of issues, right? Starting from like taste, color, to like that has to be masked, go to your body, right? So dissolution, disintegration should be uniform. So bioavailability depends on that, right? PK should be matching. So pharmacokinetic profile should be matching, right? PK of the excipients and the drug should balance, right? पेसेन्ट सेन्ट्रिक पनि हुनु पर्यो ड्रग ड्रग इन्टरेक्सन हुन्छ कि हुँदैन राइट त्यो डोजेस कुन डोजेस फर्ममा दिने कुन राउट अफ एडमिनिस्ट्रेसनमा दिने र कस्तो खालको पेसेन्टलाई कुन भ्यालुएबल हुन्छ जस्तै एउटा बिहोस भएको मान्छेलाई ट्याब्लेट बनाएर त भएन वी हेभ टु इन्जेक्ट देम राइट तर हेल्दी मान्छेलाई घरमा बस्ने मान्छेलाई फेरि इन्जेक्सन दियो भने त यु हेभ टु गो टु हस्पिटल एभ्री टाइम सो वाट डोजेस फर्म इज बेटर फर द पेसेन्ट डिपेन्ड्स अन द टाइप अफ थेरापेटिक इन्टरभेन्सन यु वान्ट टु गिभ राइट सो फिनिश प्रडक्ट कति स्ट्रेन्थ कति स्टेबल कति वर्षमा सेल्फ लाइफ कति छ सो देयर इज अ लट गोइङ behind the scene in drug discovery and I want to really appreciate the field when you talk about drug discovery and development people typically miss these two people process chemist and formulation scientist but they are the backbone of drug discovery and development wouldn't complete without their contribution right कसैलाई एक टन अफ ड्रग्स बनाइदिएन भने पनि जाँदैन र फर्मुलेसन प्रपर दिएन भने पनि द पिल एपिआई इटसेल्फ इज युजलेस until we can do what formulation scientists do. So there is also a very nice field waiting for you guys wherever you want to work. But I'm just trying to point out different areas so that you can be motivated, find your own niche, and think about this in a deeper level. Okay, I'm not a formulation scientist and I'm not a process chemist. So there are a couple of regulatory things too. So there are files of IND, investigational drug component here, right? And NDA, new drug application component before. So I have already walked through this process, but I also want you to be aware that there are regulation too. And there are, for you guys, there are regulatory sides too, right? A company make drugs, but there are regulatory side that should be testing that the drug is efficient or not. So like DDA in Nepal, right? So regulatory side is also very important in drug discovery and development. Now, the last but not the least is clinical trial. Okay, so there is a lot to learn in clinical trial, but for your level, I think you have to. It's important for you to know what happens, what goes in these trials, right? So preclinical is also called zero. In phase 1, it is done on safe, healthy human volunteers just to assess if the compound is non-toxic. It's mainly to assess how the compound is behaving, and it is done in a very less number, average of 50. And then the second is safety and dosing. It's actually done in diseased patients, right, with their consent. The participants is around 200, and यसले चाहिँ दबाईले काम गरिरहेको छ कि छैन औषधिले काम गरिरहेको छ कि होइन भनेर थाहा हुन्छ Third is a big multi-center trial run across different country. Race, caste, gene, सबै कुराहरूलाई चाहिँ एउटा ओभरअल स्टडी दिन्छ. It gives an idea about world भरिको मान्छेलाई यो बिरामीले निको पार्छ कि पार्दैन. And then post marketing surveillance, post approval phase 4 भनेको चाहिँ once the drug hits the market, त्यसपछि पनि त्यसको रिसर्च गर्न छोड्नु हुँदैन because you know there are a lot of drugs that has been pulled out of the market, right? I think that would be a good question for you guys too, because they don't meet the label. Some had a long-term effects where so while these trials were going, so many things got missed out. पछि त्यो कुरा देखियो and they have to be pulled out. So post marketing surveillance is also equally important. And okay. Now, this is a question for you guys, right? Where do you want to see yourself? If you want to contribute to the journey of drug design and discovery, you want to be a molecular biologist or pharmacologist, the job of them in drug discovery is to either find early drug targets or to feed or to do molecular pharmacology in animals and try to understand what's going on. Molecular pharmacologists, biologists also try to understand the mechanism of the drug, which I haven't discussed today because drug design doesn't cover that component, but it's also important to know how drug is functioning in the body, and that's also a molecular pharmacologist's job, right? Medicinal chemists are the central players in drug discovery. They typically work on design and synthesis, but they have to be able to interpret data from each sector and make a smart decision, because at the end of the day, the compound is a chemical substance, and medicinal chemistry can think rationally about a chemical structure that can play in multiple facets, right? Process chemist, the job market is never dying for them, because you have to make tons and tons and tons of kilograms of material during clinical trial or after. And I really appreciate the field, because without them, the drug wouldn't reach to people. Formulation chemists, formulation scientists do a tremendous job in making API to a valuable product that's called medicine, a furnished product that a patient can take. And there is a lot of science, knowledge, and effort that goes into it. So that's a very beautiful field too. So there are regulations, as I suggested, and there are scientists in a clinical trial, physicians, nurses, you name it. There are many. Okay, so there are a lot of places where you can see yourself in the future if you choose to pursue a career in pharmaceutical sciences. Okay, a beautiful career is waiting for you, and I want to wrap this up with a line saying, "Do you want to be a superhero?" Because making a medicine is the task of superhero, and do you really want to be Shaktiman? So which factors should be considered from bachelor level to pursue master's in medicinal chemistry? So I have added this just to justify to all the audience effectively. So this is the question asked. It's a very good question. I can't answer for other countries, but I can answer for United States. Um, if you want to come to United States to pursue a master's or a PhD degree, number one thing is you need to work on your CV. You need to make your profile very, very powerful. So I can suggest some of the ways. So every individual has their own ways to make their profile powerful. Number one, so I can suggest you some that I considered while I was applying. So number one, I would say is to be to have some research experiences or intern experiences, right? So during your 4 years, so when you want to come as a formulation chemist in to pursue your master's degree or PhD in formulation chemist, the skill that people are looking for you is analytical skills. People wouldn't be crazy about making tablet or capsule because in the research, that's a very old thing, right? Research grows, but they really value your analytical skill. If you want to come as a medicinal chemist, they wouldn't value your design skill. Rather, they would value your synthetic skill. So if you have an opportunity to work for someone, learn synthetic skill, that would be really valuable. One thing that you can add, all of you, to your toolkit is use of this computational technique, right? So there are free version of Schrodinger equivalent tools that are available, that you can use for free and do a lot of docking, MMGBSA calculations. So for example, you can pull a database, pull a protein of your interest, dog them. You can do a lot of this calculation that I was saying, pharmacokinetic value, ADME profiles in silico, and you can publish that paper actually. I mean, it's going to be a good story if you just do computation, and when you are applying to US, you can say, well, I have experience in solving this target with this database, and these were my rationale. So if there is a good person, computational trainer for you, do an internship for the person or do some research experience. So these are the ways that really keep you above. Okay, that's my first tip. Have those values. The second tip I want to say is be involved in different things like leadership qualities, right? Be involved in presentation, be involved in participation, organize a conference or take a part in conference, be a speaker. So just show your leadership skill. Third is be very nice and humble person, right? So these are the personal traits that they are very quick to find and they are very good at. So when you are interviewing to one of the professor or department, be very nice. Don't ask questions that make you that they feel they perceive as a bad question. Okay, just be humble, be polite, respect everyone. Okay, so have that trait too. Fourth, GPA, though that's least important. Having good GPA is an easy way. However, you wouldn't guarantee. So high GPA and every qualities I've asked you, try to instill that from today. The last thing would be a good statement of purpose, letter of recommendation, all those things. But that comes when it's time for application, and it doesn't take much time. But the time that I'm saying you will take a lot of time. So if you are really thinking US as your career, kindly consider these tips, and when people see your profile, they will be impressed, and I think your dream is right there. So here I think I end my talk. Drug design, a story of successful medicine from bench to bed. I'm happy to answer any question you might have for me. Are you running out of time?
No, sir, we are actually on time right now. So I would like to thank you for this nice story or presentation. I guess you have roused the superhero in as each and every one of us, which was hidden. So let's begin to Q&A. Here are some questions I would like to read out for you.
Sounds good. It would be great if I can read when you are reading too.
Okay, sir.
Let me try. Okay.
Okay, sir. First of all, Khum Bahadur Bhandari has asked why we
Uh-huh.
cannot use recent data despite of 2008. I
Oh,
guess it's
okay.
the first title.
So yeah, because yeah, that's a great question. Recent data would be great. So there are two reasons for this. So the recent data for how long drug takes is same, right? If we consider the time before the advent of AI, there's no data that is saying drug discovery and development takes 5 years. But that data is a complete picture of what is reflective human today. Therefore, that data has been pulled. Though I mentioned during my talk that it's promising that AI can bring down the time, but it's not guaranteed, it's not shortened. So that data is still a reflection of the current scenario. So yeah, thank you for the question though.
Khum Bahadur Bhandari has asked another question. That is the development of COVID vaccine is structure based or ligand based. Can you explain? Is there any role of drug discovery and development?
Okay, so vaccine development is not covered today. Vaccine development is different than drug discovery and development. So, vaccine development trains your immune system to produce antibodies when actual interaction with antigen happens, right? So, in the drug development, it's very different. You can target immune system in drug development too, but it's going to be a target. Whereas in vaccine development, you administer something, it can be mRNA, it can be dead vaccine, dead disease, dead virus, bacteriophage, but you are triggering your immune system to produce an antibody in the memory cell. So that will be circulating in your lymphocytes. And when it really sees that trigger, the pathogen, it does antigen antibody reaction to collapse that. So it was beyond the scope of today's talk. However, I appreciate the question and enthusiasm.
Thank you, sir. The next question is asked by Nilu Regmi. She has asked, is top list scheme mandatory nowadays for designing a drug?
Is what?
Top list scheme.
What
Top
is
list scheme.
that? I might not be aware.
Okay.
So why can't I read the chat? Let me see. Huh? Okay, I can read now. Okay, what is top list scheme? I don't know what that term is. Okay, where is Nilu? Can you explain me this? Okay. Yeah, we can move on. I'm sorry. I don't.
Okay,
I'm
sir.
not aware of this term exactly.
Okay, sir. Then Ramesh Podal has asked, how safety is assured or predicted in the early phase of drug development?
Yeah, that's a very good question. So safety is really important, right? So when you identify a heat compound, moving to a lead compound, the first thing we do are AIMS test. If you have not heard it, it's a mutagenic test. It tells about carcinogenicity and mutagenic property. We treat the compound in different cell lines, healthy cell lines, for example, kidney cell, lung cell, heart cells, right? And then we establish that the compound is not killing those cells. The compound is only killing the target. The compound is only killing the protein that is expressed, not other things. So that's how we establish safety. And it's so important to do that early on because you don't want to later realize that, oh, your compound was genotoxic actually. So that's a very good question. And we take very precautionary measures to ensure that our products are safe moving forward in every stages.
Next, the question is asked by Newton Sual. He has asked, how can drug resistant mechanism, example, efflux forms, mutation-driven resistant, be overcome through rational drug design?
Yeah, that's a very good question too. Right. So mutation driven resistance is random. It's very tough to predict. If it is scientific, if it is systematic, then there is a chance. However, if flux pump has a distinct mechanism, right, how it speeds things out. So to combat that, the drug developed should be able to bypass the efflux pumps. So efflux pumps has certain properties that it spits out drug when it is trying to enter the membrane. So you need to bypass those properties, because efflux pumps at the end of the day is also going to either bind through active transport, or by diffuse through a passive transport. So one of these two, and there are certain parameters. So people have developed a lot of strategy to move away from this problem with efflux pump. But very nice question. So mutation driven resistance, though, if it is random, there is no way to solve it, right, by a rational design. However, if it's predicted, so if this resistance is going to come, if it's predicted, there's a way you can tackle if it is actually in the active side of the protein pocket. You can change the structure of the dog to target the mutated protein. However, that's not very common strategy, employed in the drug design and development. Very good question, though.
Next question is asked again by Ramesh Podel. He has asked, how did you initially decide which portion of the lead compound needs to change to improve safety? Or was it just hit and trial approach?
How did you initially decide which portion of the needs to change to improve safety? Okay. Which portion? Yeah, okay. That's a great question. So there are two things I would like to say. So there are data and there is a chemist rationale. Okay. So for example, so by data I mean you take your compound and you see what happened to the compound, right? So what was that toxicity issue? So the toxicity could be the compound itself, the metabolite, and what it is attacking. It is attacking DNA, it is attacking some other protein, right? Once you identify that, you can isolate that and see the structure. And now you know, okay, this is the most electrophilic side of the protein, that's hydroxyl. So the most nucleophilic side was hydroxyl group, or I have epoxide in my compound, that was most electrophilic side. Once you address that, you want to tune the reactivity of that. So it's very guided and very scientific, not hit and trial. However, hit and trial sometimes could be beneficial too. When you couldn't figure out what is actually happening, you change a lot of things and try to solve it. However, in the current world, people want an answer of why it is toxic. Try to understand a chemical rationale for that and try to fix it via rational drug design.
Okay. Next question is asked by Dinesh Chaudhary. He has asked, how to decide the doses of a specific drug?
So by doses, you mean dose amount
Doses
or
from,
the formulation?
I guess doses from formulation.
Formulation, okay. So I'm not a formulation chemist, so I might not be best in answering this. Maybe Govinda would be, but so the doses of a specific drug is so first of all, what are the target diseases, right? So for most of the disease, our goal would be to develop an oral formulation, at least a pill. So because it's stable, right, more stable than liquid. However, oral form is great. So that is what we typically tend to do when we want to develop something for a common disease. However, for something for, like as I said, anesthesia, right? We can't develop oral. We have to develop that. But if you are asking dose, there is a very systematic way of finding dose. On like when you do in experimental animal, you have to find a lethal dose, effective dose, and there is a formula that will give you the actual dose. And when you go to human, there are definite calculations that you can apply to find out the dose of the compound. But deciding doses is that. But when you fail to establish that the compound can't be given orally, then you go, what is the second easy way? And then you might go to a parenteral route or intravenous route or intramuscular route. It depends on a lot of factors. But the first priority would be oral for sure.
Next, the question is asked by Kiran Sapkota. What about drug regulatory affairs, sir? Is it related with medicinal chemistry?
Guys, I'm so excited that you have such a nice question and a lot of questions. That's very good. I'm very happy. So what's about drug regulatory affairs, or is it related with medicinal chemistry? So there are two sides, right? One side says we made the best drug. We made the drug, you give it to the patient the patient will be cured. Because those are investors. Every pharmaceutical industry are investors. They are business ventures. They are making drugs because they are serving mankind, but they are also making money. So who is going to say no? This is a bad drug versus wow, that's a great drug, right? There is a regulatory affairs like us, right, who works on the principle of a drug efficacy. So they just see the data and they say, well, this is good, this is not good, it's too toxic, you have to bring it down. So there are a lot of things that happens in IND and NDA that the data they see and say it's not good. So there are people who needs to approve or disapprove that, right? So the knowledge of medicinal chemistry is crucial in drug discovery. So those people are highly effective, and because of that, the FDA agencies like FDA exist. So there is a huge role to be played by medicinal chemistry in the regulatory aspects too. Hopefully, I answered your question.
Next question is asked by Newton Suwal. He has asked, what computational approaches can be used to model the crosstalk between multiple signaling pathway in cancer and identify potential drug targets?
Yeah, that's a very good question. So target is one, but the crosstalks in the target is the other way, right? How a protein and protein talks, how a cell and cell talks. So they are also signaling pathways, right? When you overexpress something, so when a agonist come and bind to the protein, that sends a signal to the other protein to express. So there are also crosstalks. So that we can that we can identify. So there is a mechanism that happens, right? For example, even this voltage guided channels, right? That's also a crosstalk in a different sense. So once you identify those patterns, they can be regulated. For example, what is the agonist that is coming to bind, and what's the signal that is given to the other side of the protein? Once you understand those patterns, we can either tune the signal, we can either tune the activation. So there are a lot of things on the cascade that we can play too, not only on the target, but that's also a very good question. So targeting the cascade, targeting crosstalks are also important. Can I speak
Yes,
with her? I
Kiran.
have some questions.
Yes,
Yeah,
yes,
go for
please.
it, Kiran.
Hello, sir. Morning.
Good morning.
म नेपालीमै बोल्छु है त।
मजाले।
मेरो नाम किरण सापकोटा। म भर्खरै ब्याचलर इन फार्मेसी पूर्वाञ्चल युनिभर्सिटीबाट कम्प्लिटेड गरेर बसिरहेको छु।
हजुर।
अनि अनि पूर्वाञ्चल युनिभर्सिटीबाट फार्मेसी बिफार्म फार्मेसी सकेर अहिले बसिरहेको छु।
हजुर।
अनि मलाई यहाँ ठ्याक्कै देखेर तपाईँको वेबिनारमा जोइन हुन पाउँदा एकदम खुसी पनि लागेको छ। अनि म मेनली चाहिँ ड्रग रेगुलेटरी एफेयरको बारेमा अलिकति रिसर्च गरिरहेको छु है।
हजुर। हजुर।
अनि यो चाहिँ यो मेडिसिनल केमिस्ट्रीसँग कति रिलेटेड छ होइन। अनि हामी मास्टरमा आउँदाखेरि अब म पनि युएसकै लागि कुनै कुनै युनिभर्सिटीहरू यसो रिसर्च गरेर बसिरहेको छु। अनि
हजुर।
त्यसमा हामीले आउन सक्छौँ कि सक्दैनौँ यहाँबाट हामी कतिको।
मजाले सक्छौँ। हजुर, तपाईँहरू जस्तै मान्छे नै हो आउने होइन। सो इट्स अ पुल अफ स्टुडेन्ट्स, स्मार्ट स्टुडेन्ट्स नै आउने हो। सो मजाले सक्छौँ टु एन्सर द्याट। तर त्यो आउनलाई चाहिँ यु हेभ टु वर्क टुवर्ड्स द्याट डिरेक्सन। सो अर्को पनि सब्जेक्ट हुन्छ, रेगुलेटरी सब्जेक्ट पनि हुन्छ फार्मेसीमा होइन। रेगुलेटरी अफेयर्समा रेगुलेटरी सब्जेक्ट पढेको मान्छे पनि हुन्छ, ड्रग डिस्कभरी पढेको पनि हुन्छ, मोलिकुलर बायोलोजी पढेको पनि हुन्छ। सो इफ युर इन्ट्रेस्ट इज एफडिए, there are a lot of jobs there too, in research, in regulation, in being a member of the panel, a candidate. So yeah, धेरै ठाउँमा चाहिँ स्कोप छ. So that is also like an organization, अर्को organization. So that's just there to check versus the organization that is to make drug. Okay, so त्यति मात्रै फरक हो. They talk with same science, they have same expectation for the drug. Knowledge is same.
अनि मलाई अर्को पनि के कुरा जान्न मन लागेको छ भने तपाईँले अब तपाईँ पिएचडी गर्दै हुनुहुन्छ तपाईँ प्युर्डु युनिभर्सिटीमा होइन। त्यो युनिभर्सिटीमा चाहिँ हाम्रो ड्रग रेगुलेटरी एफेयर भन्ने कोर्सहरू एभाइलेबल छ कि छैन। लिमिटेड कोर्स मात्रै, लिमिटेड युनिभर्सिटीले मात्रै अफर गर्दो रहेछ भनेर मैले रिसर्चबाट बुझेको थिएँ।
पड्युमा छ, पड्युमा चाहिँ त्यो डिपार्टमेन्टको नाम चाहिँ फार्मेसी प्र्याक्टिस भनेर छ होइन।
हजुर हजुर।
फार्मेसी प्र्याक्टिसमा चाहिँ ड्रग रेगुलेसनको कुरा हुन्छ एक्चुअली।
हाम्रो यहाँको बिफार्म इक्विभ्यालेन्ट हुन्छ नि सर ड्रग रेगुलेटरी एफेयरमा
मजाले।
त्यही त। अनि कुनै कुनै युनिभर्सिटीहरूले इक्विभ्यालेन्ट पनि अब खोज्दो रहेछ। अनि हाम्रो क्रेडिट आवर पुग्छ कि पुग्दैन त्यो अनुसारले मात्रै एडमिसन इनरोल गर्छ भनेर
4 वर्षको
भनिरहेको
ब्याचलर्स हो नि होइन।
हजुर हजुर 4 वर्षको
4 वर्षको ब्याचलर्स सबैको पुग्छ, डोन्ट वरी अबाउट इट। हाम्रो बढी हुन्छ क्रेडिट यिनीहरूको भन्दा। 3 वर्षको ब्याचलर्स भयो भने मात्रै प्रब्लम हुन्छ। 4 वर्षको ब्याचलर छ भने इक्विभ्यालेन्ट माग्यो भने चाहिँ यहाँ धेरै इक्विभ्यालेन्ट गराउने हुन्छ। मेनली WES चाहिँ स्ट्यान्डर्ड मानिन्छ, त्यहाँबाट इक्विभ्यालेन्ट गराएर पठाउँदा हुन्छ। तर कसैले हामी नेपालको डाइरेक्ट ट्रान्सक्रिप्ट पनि मान्छ।
ए सर त्यही त। अब मैले पनि अब अहिले मतलब भर्खरै एउटा रिसर्च पब्लिस गरेको छु, तर मैले क्लिनिकलमा गरेको छु होइन। अब यसले भर्खर तपाईँले एक्सेप्ट गरे पब्लिस चाहिँ भएको छैन इन्टरनेसनल
हजुर
जर्नलमै। अनि
हजुर
त्यता गरिरहेको छु। अब मलाई चाहिँ मेनली अब फार्मास्युटिकल रेगुलेटरी एफेयरहरूमा सबै हेर्न मन लागेर रिसर्च क्लिनिकल रिसर्चहरू पढ्न इच्छा लागेर चाहिँ तपाईँसँग यसो सोध्नको लागि नि सर।
मजाले मजाले यस्तो गरौँ, सबैको टाइम पनि छ होइन। सो
हजुर
मेरो मेरो म यहाँ मेरो इमेल लेखिदिन्छु होइन च्याटमा। मलाई
हजुर
इमेलमा मजाले जे सोद्धा पनि हुन्छ। आई एम भेरी ह्याप्पी टु एन्सर यु गाइज होइन। म पनि कुनै पोइन्टमा चाहिँ तिमीहरूकै जस्तो स्टेजमा थिएँ। सो आई रियली फिल यु, स्पेसली फार्मेसी पढेको मान्छेहरूसँग। सो
गुड मर्निङ सर।
गुड मर्निङ
सर मलाई एउटा क्वेसन रहेको छ। जब
हजुर
हामी बोर्डर छेउमा नै बसिरहेछु नेपालको, अनि
हजुर
के हुन्छ
हजुर
मार्केटतिर जाँदाखेरि त्यो इन्डियन मेडिसिन राम्रो काम गर्ने, नेपाली मेडिसिन राम्रोसँग काम गर्दैन, त्यस्तो किन भएको होला? जब बन्ने प्रक्रिया होइन फार्माकोपियाबाट नै बन्ने हो होइन। नेपालमा
हजुर
धेरै जसो मेडिसिनहरू इन्डियन फार्माकोपियाबाट नै बनिराखेको हुन्छ युएसए, तर इफेक्टिभनेस किन कम बेसी हुन्छ?
ओके, दिस इज अ भेरी गुड क्वेसन होइन। सो एक्टिभ फार्मास्युटिकल इन्ग्रिडियन्टको रिजेक्सन हुन्छ नि जुन प्युरिटी प्रोफाइल, त्यो भनेको चाहिँ एकदमै स्ट्यान्डर्ड कुरा हो कि। तिमीलाई म एउटा इक्जाम्पल भन्छु। 99.9% कुनै कुरा प्योर छ र कुनै कुरा 99.99% प्योर छ भन्नु भनेको 10 टाइम्स बढी हो कि। यदि तिमीले 99% कुनै कुरा बनायौ, त्यसलाई 99.9% बनाउन तिमीलाई 10 टाइम बढी लाग्छ। सो 10 प्यारासिटामोल 100 रुपियाँ पर्यो अब है। अर्को एउटा 99.99 थप्यो भने 10 टाइम्स बढी पर्छ, 1000 रुपियाँ पर्छ हो। सो जति एक्टिभ फार्मास्युटिकल इन्ग्रिडियन्टको प्युरिटी जति बढायो, जति ओभरअल फर्मुलेसनको प्युरिटी बढायो, र जति यो के भन्छ यसलाई प्यारामिटरहरू हुन्छ कि टक्सिक प्यारामिटरहरू जति कम गरायो, त्यो ड्रग त्यति इफेक्टिभ हुने हो है। सबसे इम्पोर्टेन्ट कुरा यो हो, कति प्योर छ तिम्रो कम्पाउन्ड, कति पोटेन्ट छ, कस्तो फर्मुलेसन छ होइन। सो युएसको ड्रगहरू चाहिँ सितिमिति बढी इफेक्टिभ किन हुन्छ भन्दा चाहिँ दे हेभ अ भेरी स्ट्रङ रेगुलेसन कि। यस्तो रेगुलेसनहरू धेरै ठाउँमा हुँदैन। अब नेपाल इन्डियामा पनि यही भएको हुनसक्छ, अरू केही रिजन हुँदैन यसमा। दिस इज द सेम मोलिक्युल, तर त्यो खाँदाखेरि टार्गेटमा गएर रेजिडेन्ट टाइम पुग्ने र जो इफेक्टिभ हुनुपर्छ नि त्यसको लागि त्यो पुगेको छ कि छैन, त्यसलाई बायोइक्विभ्यालेन्स टेस्टिङ गर्छ धेरै देशहरूले होइन। धेरै कन्ट्रीहरूले चाहिँ बायोइक्विभ्यालेन्स टेस्टिङबाट चाहिँ जुन चाहिँ ओरिजिनल डिस्कभर ड्रग र यसको इक्विभ्यालेन्स बराबर छ कि छैन भनेर हेर्छ। तर त्यो चाहिँ पनि हाम्रोमा धेरै प्रोडक्टमा गरिँदैन। सो देयर आर अ लट अफ रिजन्स। मेन भनेको चाहिँ टार्गेट रेजिडेन्सी, कम्पाउन्डको प्युरिटी प्रोफाइल, कम्पाउन्डको अरू यो हुन्छ नि यो के भन्छन् थेरापिटिक प्रोफाइल्सहरू नपुगेर हो। एन्ड द्याट इज रियल। कम्पनी राम्रो कम्पनीको औषधि खानु जहिले पनि 1-2 रुपियाँको लोभ नगर्नु, सजेसन मेरो।
सर त्यही अब मैले नि अहिले मेडिकल रिप्रेजेन्टेटिभतिर अहिले काम गर्दैछु, अनि डक्टरहरू पनि अब होइन उहाँले के होइन के मलाई नै सोध्छ कि नेपाली कम्पनीमाथि विश्वास लाग्छ? मैले हो सर विश्वास लाग्छ नि, कैसे मलाई त लाग्दैन नेपाली कम्पनीहरू कामै गर्दिन। म चाहिँ त्यही मेडिसिन मैले 10 ml दियो भने काम गर्छ, अनि नेपाली मेडिसिन दिँदाखेरि 15 थप्नुपर्छ, 20 थप्नुपर्छ, अनि त्यतिको इफेक्टिभ हुन्छ भनेर। अब त्यसलाई अब एज अ फार्मासिस्ट भन्नाले त अब सबै त फार्माकोपियाको हिसाबले जबसम्म त्यो फार्माकोपिया होइन यति पोटेन्सी हुन्छ अनि मात्र त्यो पास गर्नुपर्छ नि क्युसीबाट, क्वालिटी कन्ट्रोलबाट होइन। त्यो अनुसार त अब सेम तरिकाले नै बन्ने हो, तर इफेक्टिभनेस त्यो डिफ्रेन्ट भइहाल्छ। अब कसरी जवाफ दिने त्यही त्यो भइहाल्छ सर।
आई थिङ्क यो एन्सर मेरो स्कोप भन्दा बाहिर भयो है।
म चाहिँ यसमा के कुरा भनौँ ल,
हजुर
कविन दाइ
के बुझ्नु पर्यो भन्दाखेरि चाहिँ
हजुर
जस्तै फर्स्ट कुरा भने म
मल्टिपल
कन्टिन्यू गरौँ, मैले एन्सर दिन सक्छु यसको किन महँगो सस्तो पर्छ, राम्रो नराम्रो भन्ने कुरा।
कविन दाइ
नभए तपाईँले पनि हुन्छ।
कविन दाइ ल
एउटा चाहिँ सेफ्टीको कुरा हो होइन। जुन प्युरिटीको कुरा आएको छ, हामीले जहिले पनि औषधिमा दुईवटा कुरा हेर्छौँ, एउटा इफिकेसी एउटा सेफ्टी होइन। अनि सेफ्टी चाहिँ हाम्रो फार्माकोपिया भनेको एउटा बेसिक रिक्वायरमेन्टवाला डकुमेन्ट भयो होइन। नेपालमा चाहिँ एज अ बाइबल एउटा मेजर स्ट्यान्डर्ड बुक भनेर नै फलो गरिरहेको छौँ, तर त्यति मात्रले पुग्दैन होइन। त्योभन्दा अरू थुप्रै रिक्वायरमेन्ट्सहरू फुलफिल हुनुपर्छ जुन बाहिरतिर हुन्छ, नेपालमा या इन्डियामा चाहिँ हुँदैन है। अब प्युरिटी अभियसली 99.9% र 99.99%मा धेरै नै सस्तो महँगो कुरा आउँछ। हाम्रो फार्माकोपियाहरूले 99.9 भन्छ भने विकसित देशहरू या विकसित औषधि कम्पनीहरू चाहिँ 99.99% प्युरिटी युज गरेको हुनसक्छ है, त्यसले कस्ट फरक पर्छ। इभन हाम्रो यो डिजोल्युसनहरू हुन्छ होइन, डिजोल्युसन हामीले एउटा मात्र डिजोल्युसन मिडियाबाट पास गरेर ल पास भयो भनेर चाहिँ हामीले मार्केटमा रिलिज गरिरहेको हुन्छ भने नर्मली प्रायः जस्तो चाहिँ मल्टिमिडिया 4-5वटा डिजोल्युसन स्टडी डिफ्रेन्ट पिएचमा डिफ्रेन्ट डिजोल्युसन मिडियामा गरिरहेको हुन्छ र त्यसको आधारमा बसेर फेरि फर्दर चाहिँ हामीले बिएबी भन्छौँ, बायो एबिलिटी बायो इक्विभलेन्सको स्टडीहरू गर्छौँ, त्यो फेरि अर्को कस्ट लाग्ने भयो होइन। अनि त्यसपछि अर्को प्युरिटीको कुरा गर्दै गर्दाखेरि जुन प्रडक्ट डेभलप गर्छौँ, प्रडक्ट डेभलप गर्दाखेरि जस्तै युएसमा चाहिँ एलिमेन्टल इम्प्युरिटिजहरू हेर्ने गरिन्छ होइन। त्यसमा अब मेटल्सहरू अहिले लिस्टेड हेभी मेटल्स भन्दा बाहेकको अरू मेटल्सहरू पनि हेर्ने गरिन्छ, प्लस रेसिडुअल सल्भेन्टहरू। नेपालको केसमा चाहिँ रेसिडुअल सल्भेन्ट फ्यु फार्मास्युटिकल कम्पनीले चाहिँ हेर्न सुरु गरिसकेको छ, जसमा चाहिँ ग्यास क्रोमेटोग्राफीहरू छ, त्यसमा गरिसक्यो। तर स्टिल हामी चाहिँ त्यति रेडी चाहिँ छैनौँ। प्लस अरू इम्प्युरिटिजहरू पनि हुन्छ होइन। अब यो मेडिसिनल केमिस्ट्रीमा अझै बुझिन्छ यो कुराहरू। इम्प्युरिटिजहरू एकदम यति सानो मात्रामा हुन्छ, कहिले कहिले चाहिँ हाम्रो एनालाइटिकल इन्स्ट्रुमेन्ट्सहरू एक्जिस्टिङ एनालाइटिकल इन्स्ट्रुमेन्टले डिटेक्ट गर्न नसक्ने अवस्था पनि हुन्छ। भनेपछि एनालाइटिकल इन्स्ट्रुमेन्ट राख्ने बित्तिकै नेपाल जस्तो देशको एकदम महँगो कस्ट बढ्न सक्छ। जस्तै अब हामीले अहिले एसपिएलसी युज गरिरहेछौँ भने हामी अब मास स्पेक्ट्रोस्कोपीतिर गइसक्यो होइन, तर नेपालमा चाहिँ त्यो प्र्याक्टिसमा आइसकेको छैन। भनेपछि यस्तो यस्तो चिजहरू चाहिँ कस्टहरू बढ्दै जाने हो र यस्तै चिजले चाहिँ काम गर्छ गर्दैन भन्ने कुरा चाहिँ हुन्छ। अनि हाम्रो एज अ कन्ट्री पनि रेगुलेटरी सिस्टम चाहिँ डब्लुएचओले चाहिँ लेभल 1मा राखेको छ भनेको, लेभल 0 भनेको रेगुलेटरी सिस्टम केही पनि छैन भन्नु हो, र लेभल 5 भनेको एकदम मेच्योर भइसक्यो भन्नु हो। भनेको विकसित देशहरूको फार्मास्युटिकल रेगुलेटरी सिस्टम चाहिँ एकदमै मेच्योर भइसकेको छ र हाम्रो जस्तो देश चाहिँ रेगुलेटरी सिस्टम जस्ट सुरु गऱ्यौँ र जस्ट रेगुलेसन गर्ने स्टेजमा छौँ भनेर चाहिँ डब्लुएचओले पनि लेभल 1मा तोकेको छ। भनेपछि हामी अझै मेच्योर हुनलाई लामो समय छ। त्यो भएको भएर एउटा डाउटको ठाउँ चाहिँ हुन्छ र त्यो भन्दै गर्दाखेरि चाहिँ इन्डियन कम्पनीले काम गर्छ, नेपाली कम्पनीले काम गर्दैन भनेर चाहिँ एकदमै जनरलाइज र ब्ल्याङ्केट एप्रोचमा चाहिँ भन्न चाहिँ मिल्दैन। मैले चाहिँ यति आन्सर गरेँ होला सायद।
हो, त्यस्तो भन्न मिल्दैन। यु कान्ट से इभन अमेरिकन कम्पनी राम्रो उहाँको नराम्रो भन्न मिल्दैन। यु निड टु फाइन्ड आउट वाट साइन्स इज रेस्पोन्सिबल फर द्याट होइन। एनिवेज गाइज, आई थिङ्क आई गो now.
I have also one question. Someone has asked anonymously. What do you suggest to someone who is frustrated mailing professors for almost 6 months now, for like 200 to 300 US professors? So I guess someone is asking for suggestion.
So actually, question Bistari Vanada. What is suggestion? Okay.
What do you suggest to someone who is frustrated mailing professors for almost 6 months now? I guess he or she has mail around 200 to 300 US professors and asking a suggestion.
Wow, yeah, that's like good luck. Yeah, that's a good approach to start with. होइन 200-300 जना प्रोफेसरलाई इमेल गर्ने मान्छेको मोटिभेसन क्लिन देखिन्छ होइन। सो द्याट्स गुड। तर म एउटा कुरा के भन्न चाहन्छु भन्दा चाहिँ प्रोफेसरलाई मेल गर्नु मात्रै विन होइन। सो दुईटा कुरा गर्नुपर्छ। प्रोफेसरलाई मेल गर्दा चाहिँ आफ्नो इन्ट्रेस्ट पहिला लेख्नुपर्छ, एउटा पावरफुल इमेल बनाउनु पर्छ, प्रोफेसरको रिसर्चसँग ट्युन हुने, आफ्नो एक्सपिरियन्ससँग बाँधिने र आफ्नो प्यासनसँग जुझ्ने। सो यु हेभ टु थिङ्क 3 थिङ्स। ओके, वाट आई एम गोइङ टु डु माई पिएचडी इन, इज द्याट प्रोफेसर द राइट मेन्टर टु ट्रेन मी अन द्याट फिल्ड, एन्ड इफ आई गेन माई पिएचडी विल आई डु रियली ग्रेट। यो 3टा क्वेसन सोचेर इमेल लेख्नुपर्छ। हरेक इमेल सुड बी युनिक टु अ प्रोफेसर। दोस्रो कुरा, प्रोफेसरको रिप्लाई आयो भने ग्रेट, रिप्लाई आएन भने एप्लाई गर्न कहिले छोड्नु हुँदैन। सो दिस मैले कतिजनालाई यो सजेसन दिइरहेको हुन्छु कि दाइ प्रोफेसरले इमेल गरे आएन, जस्ट किप एप्लाइङ। एप्लाई गर्दा चाहिँ 2-3टा टप टियरको स्कुल, 2-3टा मिडल टियरको स्कुल, 2-3टा लो टियरको स्कुल, एउटा स्कुलको ब्यालेन्स बनाएर एप्लाई गर्ने, बेस्ट एप्लिकेसन क्रिएट गर्ने, र 3rd 4th इयरमा जो छ मैले अघि भनेको कुराहरूलाई चाहिँ इम्प्लिमेन्ट गरिहाल्ने। इट्स गोइङ टु बी 1.5 आवर, बट 1.5 इयर। तर 1.5 इयरको त्यस्तो एक्सपिरियन्स इज गोइङ टु बी सो भ्यालुएबल कि होइन। त्यही भएर चाहिँ त्यसले तिमीहरूलाई धेरै माथि लान्छ। इभन युएसकै कम्पिटिसनमा पनि माथि पुग्छ कि इफ यु रियली नो हाउ टु युज। कविताले भने जस्तो एचपिएलसी, जिसिएफआइडी, जिसिएमएस इन्स्ट्रुमेन्ट चलाएर म्याथड डेभलप गरेको छ, म्याथड भ्यालिडेट गरेको छ, कम्प्युटेसनली चाहिँ कुनै टार्गेटलाई डग गरेर कम्पाउन्ड निकालेको छ, एमएमजिबिएसए हेरेछ, पेपर पब्लिस गरेछ। सो दिज आर रियली भ्यालुएबल थिङ इन पिएचडी इन अनि अर्को पनि कुरा भने म दिन चाहन्छु त्यो कुरा फलो गर्ने जस्ट डोन्ट ब्ल्याटेन्टली इमेल बिकज द्याट डजन्ट लुक गुड मलाई पनि कति इमेल आइरहेको हुन्छ कि प्रोफेसरको लागि भनेर प्रोफेसरले कति इमेल मलाई फरवर्ड गरेको हुन्छ होइन सो इट्स लाइक इट डजन्ट लुक गुड वेन समन हेज अ प्याटर्न अनि हेजन वर्क हार्ड होइन अनि आफ्नो सिबी पनि एकदम राम्रो बनाउने जस्ट ट्राई टु बी बेस्ट अफ यु के योर सेल्फ के होइन त्यसपछि चाहिँ एप्लाई गर इमेल पठाऊ तर एप्लाई गर्न पनि नछोड कि यु डोन्ट ह्याभ टु गेट अ गुड रेस्पोन्स टु एप्लाई सो जस्ट एप्लाई सी एक्सेप्ट भयो भएन भने भएन बट देयर इज अलवेज अ चान्स यु निड टु टेक
म यसमा अलिकति थपिदिन्छु ल
ओके
यस्तो हुन्छ भन्नाले उहाँले भने जस्तै कस्तो हुन्छ भन्दाखेरि जस्तै ऱ्यान्डम इमेल पठाउनु चाहिँ के कुरामा चाहिँ इम्पोर्टेन्ट हो भन्दाखेरि जस्तै कसैले रिसर्चको स्पेसलाइजेसन चाहिँ तपाईँले एउटै युनिभर्सिटीमा दुईवटा प्रोफेसर होइन प्रोफाइल हेर्नुभयो एउटाले एउटामा स्पेसलाइजेसन रहेछ अर्कोमा स्पेसलाइजेसन रहेछ अर्कोमा स्पेसलाइजेसन रहेछ भने चाहिँ यहाँ स्टुडेन्टले के पनि गरेको देखिन्छ भन्दा तिनटै प्रोफेसरलाई म तेरो एरियामा चाहिँ इन्ट्रेस्टेड छु भनेर मेल गरिदिने कि ऱ्यान्डमली सेम कपी पेस्ट गर्ने तिनवटालाई पठाइदिने होइन एउटाको त रेस्पोन्स आउला भनेर अनि त्यो गर्दाखेरि उनीहरूको इभन खाना खाने बेलामा खाजा खाने बेलामा पनि अब काइन्ड अफ कुरा हुन्छ कि ए मलाई नि पठाइदियो पनि हुन्छ होइन कहिले कहिले त्यही भएर उहाँले भने जस्तो के चाहिँ इम्पोर्टेन्ट हो भन्दा ठ्याक्कै आफ्नो एकदमै स्पेसिफिक रिसर्च एरिया छुट्याउनु पऱ्यो जुन चाहिँ यदि उहाँले भने जस्तो चाहिँ पहिले आफ्नो त्यो ब्याकग्राउन्डमा त्यो रिसर्च भएको प्रोफाइल पनि हुनु पऱ्यो कि उनीहरूले बताउनु पऱ्यो नि त कि यो एरियामा चाहिँ अलरेडी सानो भए पनि काम गरेको रहेछ भनेपछि उसलाई एउटा फेथ हुनु पऱ्यो अनि त्यसपछि स्ट्रङ इमेल भनेको चाहिँ जस्तै इमेल लेख्ने हुन्छ नि प्याटर्न मैले अब यति वर्ष काम गरेको उसको रिसर्च इन्ट्रेस्टमा फिट हुन्छ कि हुँदैन त्यो उसको पब्लिकेसनमा आफ्नो कुराहरू फिट हुन्छ कि हुँदैन होइन अनि कति कुराहरू चाहिँ मैले के पनि रियलाइज गरेको भन्दाखेरि उसको यदि पब्लिकेसन प्रोफाइलमा गएर तपाईँले हेर्नुभयो भने चाहिँ उसले डेभलपिङ कन्ट्रिजको मान्छे लिन्छ कि लिँदैन इभन नेपालको कोही मान्छे छ कि छैन होइन अनि त्यसबाट पनि तपाईँले एउटा जेनरल आइडिया लिएर उसले तपाईँले एक्सेप्ट गर्छ कि गर्दैन भन्ने हुन्छ अब कति कुराहरूमा चाहिँ उनीहरूले डेभलपिङ कन्ट्री इभन नेपालको भनेपछि इमेलले रिजेक्ट गर्ने पनि हुन्छ होइन लाई कन्सिडर गरेर गऱ्यो भने चाहिँ बेस्ट हुन्छ र मेरो अर्को एक्सपिरियन्स भनेको चाहिँ इमेल लेख्दाखेरि तपाईँले कुन दिन लेख्ने भन्ने पनि सायद इम्पोर्टेन्ट हुन्छ जस्तो लाग्छ होइन किनकि विदेशमा पढाए चाहिँ सोमबार बिहान गएपछि मात्रै इमेल चेक गर्ने हुन्छ फ्राइडे अब तपाईँले पठाउनु भयो भने पुराली चेक थर्सडे पुराली चेक नहुन पनि सक्छ होइन त्यस्तो इमेलको टाइमिङ होइन त्यो टाइम जोन हेरेर उस गऱ्यो भने चाहिँ बेटर हुन्छ जस्तो लाग्छ
Yeah, I agree with Ramesh bro है so so just आफ्नो इन्ट्रेस्टलाई ट्युन गरेर इमेल लेख्नु सबैलाई नलेख किनभने देयर इज नो बेनिफिट अनि त्यो राम्रो स्ट्राटेजी पनि होइन तिमीलाई तिम्रो इन्ट्रेस्टको प्रोफेसरहरू धेरै छन् कि युएसमा जस्ट त्यो इन्ट्रेस्टको प्रोफेसरलाई खोज त्यसलाई लेख इन्ट्रेस्ट छैन भने पनि 3-4 दिन सोच मेरो इन्ट्रेस्ट के हो भनेर त्यसपछि एउटा ट्युन आउँछ क्या आफ्नो इमेलमा अनि होइन तर त्यो ऱ्यान्डमली लेख्न थाल्यो भने चाहिँ तिमीहरू आफै डिस्टर्ब हुन्छ कि त्यही भएर
अझ म एउटा कुरा अझ एउटा कुरा थपिहालेँ होइन तपाईँले त्यो पिएचडीको टपिक्स डिसाइड गर्दाखेरि तपाईँले के सोचेर डिसाइड गर्नुपर्छ भन्दाखेरि तपाईँले अब बाँकी रहेको लाइफ नै त्यही फिल्डमा काम गर्ने हो म त्यो क्यापेबल छु कि छैन होइन आइएम इन्ट्रेस्टेड रियल्ली म त्यो फिल्डमा बसेर काम गर्न सक्छु कि सक्दिनँ किनकि मैले त पिएचडी गरेपछि मेरो स्पेसलाइजेसन त्यही फेरि स्पेसलाइजेसन छोडेर होला पोस्टरमा अलिकति चेन्ज गर्न सकिन्छ तर एरिया नै पुरै टोटल्ली चेन्ज गर्न त फेरि सकिँदैन पनि जस्तो लाग्छ मलाई होइन त्यही भएर डिसाइड गर्ने बेलामा के पनि कुरा इम्पोर्टेन्ट हो तपाईँले इन्ट्रेस्ट सुरुमा विचार गर्ने बेलामा के म अबको भन्नाले 35 वर्षमा पिएचडी अथवा 30 वर्षमा पिएचडी सकाएँ भने बाँकीको 30 वर्ष यही फिल्डमा काम गर्नलाई चाहिँ म रेडी छु म रियल्ली यो सब्जेक्टमा इन्ट्रेस्टेड हो कि होइन जस्ट युएस जानको लागि अथवा अस्ट्रेलिया जानको लागि अथवा न्युजिल्यान्ड जानको लागि स्कलरसिप पायो भने गइदिइहालौँ न भनेर डिसाइड गरेको भन्ने कुरा पनि इम्पोर्टेन्ट छ है
Yep guys do you have more questions
सर वान लास्ट क्वेसन
युएस म एउटा कुरा भन्छु ल सो तिमीहरूलाई क्वेसन छ भने डोन्ट निड टु स्टप है मेरो इमेल मैले त्यहाँ लेखेको छु मेरो जिमेल एड्रेस यु क्यान अलवेज सेन्ड मी एन इमेल अनि बेस्ड अन युर क्वेसन वी क्यान टक लेटर पनि है सो इट्स नट द इन्ड अफ आवर सेसन एन्ड आई एम भेरी ह्याप्पी टु हेल्प यु गाइज इन द बेस्ट वे आई क्यान है कन्टिन्यू युर क्वेसन सरी भाइ
युएसमा नेपालको बिफार्मलाई फार्म डीमा इक्विभलेन्ट गराउन कसरी गराउने भनेर सोध्नु भएको छ
त्यो गर्न मिल्दैन अहिले फेरि 2004 को रेगुलेसन पछि बिफार्मलाई चाहिँ यहाँको फार्मास्युटिकल साइन्सेस इक्विभेलेन्ट डिग्री दिन्छ सो यु कान्ट प्र्याक्टिस डक्टरेट इन फार्मेसी इक्विभेलेन्ट युएसमा चाहिँ सो 2004 भन्दा सरी 3 भन्दा अगाडिको बी फार्मेसीलाई चाहिँ फार्म डी चाहिँ एमएलईको इक्जाम दिएर हुन्थ्यो भने अहिले चाहिँ हुँदैन सो आवर डिग्री इज Bachelor's in Pharmaceutical Sciences, which is equivalent to B Pharmacy in Nepal. Sir, हजुर आफ्नो इमेल आइडी सेन्ड गरिदिनु न. मैले च्याटमा गरेँ है. मैले कविन दाइलाई गरेर आएको थिएँ. सरी. Okay, sorry guys. Okay, my email is there. Okay. So केही छैन भने I think I need to go now. So अरु कोही क्वेसन छ भने चाहिँ I can take a couple of questions. क्वेसन छन्?
हाम्रो क्वेरीहरु सके के छ अहिलेको लागि कसैले पनि पोजन भएको छैन. So let's conclude this. IBF on the organization NPSA, I would like to extend our heartfelt appreciation to you, sir, for sharing your expertise and invaluable insights. I guess you have aroused the superhero in each and every one of us. As a part of your appreciation, we would like to present you with a virtual token of love, and it says, I would like my friend to present the virtual token of love.
Thank you guys, thank you. And you guys are really superhero, right? So I think you'll take that superhero out and involve that energy somewhere else.
I think I'm able to share my screen. So like we can like make the token of love to you later.
Okay.
Yeah, that's fine. That's fine, guys.
The token of love. Okay, we will mail it. Now I would like to request all the participants to open the camera and let's capture this special moment.
That's great. Kavin Dai, what's up?
हजुर पंकज ठीक छ पंकज. Thank you so much for your time है.
No problem Dai, anytime.
यो अलिकति हाम्रो अपडेट्सहरु अलि छैन नेपालको एजुकेसन सिस्टममा अनि ग्लोबल सिनेरियोमा चाहिँ त्यो अलिकति पिक्चरहरु रियलाइज हुने चाहिँ धेरै छ के सबैलाई हेल्पफुल हुन्छ. अनि अहिले जुन ट्रेन्ड छ त्यसले नै गइरहेको छ नि ओभरअल फार्मास्युटिकल इन्डस्ट्री नै.
हजुर
त्यो अपडेट भयो इभन हाम्रो पनि आफ्नै स्कोप्स कहाँ सक्छ कहाँ हुन्छ त्यो ठक्क थाहा हुन्छ के. अनि त्यो उसले त्यही आफ्नो करियर पाथवे त्यसरी लाइक ग्रो गर्न सक्छ हैन. त्यो चाहिँ अलिकति नेपालमा अलिकति साङ्लो छ जस्तो लागेर अनि सेसन मन्थली गर्छौं के हामीले. अहिले सायद 14th होला, सायद 14th वेबिनार.
हजुर हजुर.
सो नेक्स्ट सोचेको चाहिँ एउटा साइन्टिफिक पब्लिकेसन कसरी गर्ने भनेर सोचेको छ. अब होला नेक्स्ट सेसन अब साइन्टिफिक पब्लिकेसन्सहरुको यो अब अहिले भर्खरै क्वेशन आछ हैन मार्केटको क्वालिटीहरुको प्रोडक्ट क्वालिटीको कुराहरु त्यसको लागि गर्छौं होला सायद.
Nice. Like thank you guys, this is wonderful. Like inspiring next generation youth is really important, हैन. And you guys are doing great. मेरो चाहिँ काई पनि के हेल्प हुन्छ भने चाहिँ इन्डिभिजुअली पर्सनली फर ग्रुप आई एम अलवेज अप फर इट है त.
Okay sir, thank you sir. I would like to thank you once
मलाई पनि यस्तो म चाहिँ खास क्लिनिकल रिसर्चर हो. ल्याबमा चाहिँ बेस्ट होइन, त्यही भएर आई एम मोर फोकस अन म ड्रग सेफ्टी रिसर्चर हो कि मेडिकेसन सेफ्टी एक्सपर्ट.
हजुर
मैले अस्ट्रेलियाबाट मेडिकेसन सेफ्टीमै PhD सकायो त. सो आई आस्क क्वेसन अन द मेडिकेसन सेफ्टी हैन. त्यही भएर अब क्लिनिकल रिसर्चमा चाहिँ फेरि सबैले एकदमै टाइम कमिटमेन्ट गरेर म सबै भ्याउँछु त म भन्दिनँ. तर कोही चाहिँ मेडिकेसन सेफ्टीमा स्पेसल्ली जेनेटिक्समा चाहिँ इन्ट्रेस्टेड छ भने आई विल हेल्प.
नाइस, थ्याङ्क यू भेरी मच.
Thank you sir. Finally, it's time to conclude now. A special thank you to each and every participant who took the time to join us from different corners of pharmaceutical landscape. That's all for today. We will be back with another session of webinar. This is your host Mr. Lokmane signing out for now today. Thank you everyone.
Bye guys.
Thank you so much everyone. Thank you so much.
Thank you. Bye bye.
Thank you so much sir. Kavin sir, I think you have to end the session.
ए ल हुन्छ. Thank you so much.
Thank you sir.
है
Thank you.
ल.